Sound check, students. Hello, students. Welcome back to your own channel, Vedantu NEET English. This is Basavaraj Sir, your biology master teacher, students. I hope all of you are doing great and amazing and ready for a brand new session today. Right now, students, this is a part 2 of your principles of inheritance and variation. In the previous class, we finished the entire basics of this particular chapter. Along with the basics, we finished something called as your monohybrid cross, codominance, incomplete dominance, as well as other things was covered in the previous class, students. Now, today's goal is hopefully finishing the entire chapter today itself, right? So because today we have a lot of things to cover. We have concept of dominance, we have your star synthesis, we have chromosomal theory of inheritance, linkage and recombination, then we have sex determination, right? After that we have pedigree, mutation, and finally all the diseases. So we have a very power packed session going ahead today. Every single concept is very important. So every single concept will be followed by numericals as well as PYQs in today's class, students. So if I'm properly audible, students, if I'm properly audible and if I'm properly visible to all of you, quickly fire emoji in the chat, all of you students. Quickly fire emoji in the chat, all of you, because you are ready for genetics, right? Genetics. So today I'll be teaching you a lot of numericals. So please follow me till the very end. So students, I want all of you to promise me that you will stay till the very end of today's lecture. All of you, quickly promise in the chat. Promise. Now, so those of you who do not know, student, this is your part 1, and this is going to be the part 2 of this particular session. Part 2 of this session. Okay. And students, before I start, I want to say thank you so much, all of you. I saw the beautiful comments. On previous video and I loved it. I love the comments here. I see the dedication of the students. I see the the pure love for the subject and the bot army here. Okay, students. Now, students, let me ask all of you one simple question first of all. In my class, I always ask you questions. Very simple question. Okay, students, is it possible that you are good at every sport, or let's say you are good at every subject? Just because you are good at biology, does it mean that you will be amazingly genius in maths also, students? Is it possible? All of you tell me. Is it possible that just because you have a good memory, you are good in biology, does it mean that you'll be good in maths also, chemistry also, physics also, astronomy, astrology, everything? No, it is not possible. It is not possible that you'll be good in every field of science. All of you will have a certain specialization. Yes or no? So with that in mind right now, all of you know the concept of dominance. Let's say students, let's say there is a allele for your, let's say here we have let's take a allele from here, students. Okay, let's say here we have capital B. Here we have capital B and small b right here. Now this capital B is dominant for seed shape. Now students, tell me, what will be the phenotype of this one? The phenotype of this genotype here. The phenotype of this genotype is going to be round seed. All of you agree with me? Because this capital B is dominant for seed shape. So this dominant trait is going to be round seed. So this genotype will have this phenotype. Okay, yes or no? This genotype, students, yes, this genotype will have this phenotype here, student. This phenotype here. Yes or no? All of you? Right. Now, students, now I want all of you tell me, is it possible that this gene is dominant for every other trait? Is it possible that this gene here is, students, is my voice breaking right now? All of you? Sound check, students. All of you tell me in the chat right now, is my voice breaking, students? Sound check. Is it clear right now? Clear, breaking? One second. will be clear to all of you. Okay. Now, students, all of you tell me, is it possible that this allele here is dominant for all the traits? Yes, so is it possible? Is it possible that this allele is, let's say, this allele is dominant for round seed shape? Okay. But is it possible? Is it possible that, right, this allele is dominant for, let's say, starch synthesis also? It is not. It is not. So that is what the concept is, students. That is what the concept is here, right? So the concept is very simple, students. Just because one version of a gene, that is allele, is dominant for one trait, this capital B small b, this capital B is dominant. It is dominant for seed shape. Seed shape, it doesn't mean that it will always act dominant for everything else. Just because you are good at maths, just because you're good at maths, it doesn't mean that you will be good at every other subject. Yes or no? Simple concept. Okay. So if a gene affects more than one feature, if this capital B is also responsible for starch synthesis, if this capital B is also responsible for starch synthesis, dominance can behave differently depending upon which feature you are observing. So if you are trying to, students, if you are trying to observe the seed shape, it is dominant. But if you're trying to observe starch synthesis, it is not dominant. It is not dominant. So I hope that simple concept of starch synthesis is clear to all of you, students. I hope it is clear to all of you. Because trust me, God promise, if you read NCERT, you will not understand. Student, let me just show you, student. Let me show you right now. Just read it. Right. You try reading it. Try reading this part. It gets so confusing. All of you. Try reading this part. You will realize, student. Try reading this part. It'll be like, what is even happening? Why is it so confusing? Okay, let it load. So let it load. This starch synthesis, every other student, they don't understand only, generally. 1 second, let it load, students. My internet is a little slow. What to do? Like all of you. Almost done. Oh, finally, students, finally. Now, we'll go this paragraph right now, students. We'll go this paragraph right now. Now, I want all of you to read this. I want all of you to read this part here and see if you can understand this. This part, students, here. I want all of you to read this paragraph and tell me, are you able to understand? See this. Dominance is not an autonomous feature of a gene or the product that it has information for. It depends on as much on the gene product and the production of a particular phenotype from this product as it does not particular phenotype that we choose to examine in case more than one phenotype is influenced by the same gene. Students, tell me, students, do you understand this line? Did you understand this paragraph? No, right? Let me break it for you. It says that concept of dominance depends on how the gene product works and which trait you choose to focus on. If you focus on seed shape, it is dominant. But if you focus on starch synthesis, it is incomplete dominance there. It is incomplete dominance. That is the concept of starch synthesis. I hope it is able to understand. But see, let's say you are a good student. You are very genius. You can be a genius in maths. But is it possible that you are a genius in physics also? No. Is it possible that you have physics in, let's say, Sanskrit also? No. So depending upon which product, depending upon which field you're looking at, you can be a genius in one. Okay, students. So with that being said, students, I want all of you to tell me which is your favorite subject. Which is your favorite subject? Tell botany. Okay, students, which is your favorite subject? That is called as starch synthesis, students. Starch synthesis. Now, with that aside, students, right now, can we start dihybrid cross, students? Can we start dihybrid cross? All of you, Blessy, understand. Blessy, understand. You cannot be good at everything. Depending upon what you're trying to look for, you can be good at something. A allele can be dominant for one trait. Here, capital B can be dominant for seed shape. If you look at seed shape, we'll say dominant. But if you look at star synthesis, in this case, the same allele where it was dominant here, the same allele here can become incomplete dominant. Okay, so depending upon what you're trying to look at, the concept of dominance changes. Okay, students. Now we come to inheritance of two genes right now. Very simple, students. Very simple. I want all of you to start writing with me. Then we have a small game for all of you, students. Okay. Now, students. First thing we write is parents. Obviously, we write parents first of all. Parents. Okay. Now here, the parents are going to be what? Capital R, capital R, capital Y, and we are crossing with small r, small y, small y. Now, students, at first you will notice right now how many characters did I take here? Here I am actually taking two different characters here. This right here is going to be round, that is seed shape, and this Y is going to be yellow, that is seed color. So here in the two genes, inheritance of two genes, we take two characters. Here we have seed shape as well as seed color. So we take two characters here, Clear, all of you? Okay. This one, student, this one is small r is going to be wrinkled. Small y is going to be green color. So those are your parents, which are obviously homozygous in nature. Okay, now students, next step is called as gametes. Students, I want all of you to participate in the class. Next is going to be the gametes. Okay. Now here what do we get, students? Here we get capital R, capital Y. Here we get small r, small y. Now students, now we get F1. Yes or no? Now students, I want all of you to tell me, in this F1 we get capital R, capital R, small r, capital Y, small y. Now for this genotype, can anyone in the chat tell me what is going to be the phenotype here? What is the phenotype? The phenotype is very simple, students. Here, capital R, it has to be round. Capital Y, it has to be yellow. So round and yellow is the phenotype of your F1. Now, next step is called as what, students? Next step is called as what? It is called as selfing of F1. Okay. In selfing of F1, what do? We take capital R, capital Y, small y, cross it with capital R, capital Y, small y. We do the crossing, students. We do the crossing here. Okay, we do the crossing right now. Okay. Now, students, I want all of you to understand, when you do this crossing right, we need the gametes here. So let's make the gametes here. Okay. We have capital R, capital Y, small y. Let's try to make the gametes, students. Let's try to make the gametes here. Let's try to make the gametes. Okay. So here we have male and here we have female. Now students, how do we make gametes here? Very simple trick. This into this, this into this, this into this, and this into this. That's all. So what do we have first one? The first bakra here is what? The first bakra is capital R, capital Y. That is the first gamete. Then capital R, small y. Then small r, capital Y. Last we have small r, small y. Okay, done. Similarly, students, you need to write here also now. Write here also. We have capital R, capital Y, capital R, small y. We have small r, capital Y, and small r, small y, students. Let me show you what you need to do. You have to do cross now. So what do we get? Capital R, capital Y, capital Y. What do we get here? Capital R, capital Y, small y. Now, students, now Very simple game. Who will tell the answer first? Will I write the answer first here, or will you type the answer first? Very simple game, no, right? Very simple game. The game is very simple. Will I write the answer first here, or will you tell the answer in the comment section? Okay, shall we start? Friends, shall we start? Let me stretch. Let me just stretch a little. So shall we start? All of you. Students, shall we start? Yes or no? Yes? Shall we start the competition? Yes? Okay, wait, don't start, wait. Let me also start. I was thinking, no? Okay. Typing is fast and typing is hard, I know. That's why advantage I have. Okay. 3, 2, 1, let's go. What do we get here? Capital R, small r, capital Y, capital Y. Capital R, small r, capital Y, small y. Capital R, capital R. Students, see if I make a mistake anywhere. Catch hold of me. Small y. Capital R, small y, small y. Capital R, small r, capital Y, small y. Capital R, small r, small y, small y. What do we have here? Capital R, small r, capital Y, capital Y. Capital R, small r, capital Y, small y. Small r, capital Y, small r, capital Y, small y. Capital R, small r, capital Y, small y. Capital R, small y, small y. Small r, capital Y, small y. And small r, small y, small y. And with that, I am done. Done, students, done. Done. Okay, now, okay, fine. If you could not follow with me, it's fine. Okay. Now, let me ask you a simple question. What is the, students, what is the phenotype of this one? Anyone? It is round yellow. Let's make a round yellow, students. Let's make a round yellow here. Okay, it's round and yellow. Okay. The first phenotype which I have here is round yellow is the first phenotype here. Okay, students. This one, all of you, round yellow. Okay. This one, round yellow. This one, capital I, capital Y, round yellow here. This one, capital R, capital Y, round yellow here. But students, if you see this one, it is going to be round. But small y, students, means what? Small y is going to be what? Green, no? Here we learned that small y is always green. So can I tell this one is going to be round and green? Yes or no, students? It's going to be round and green here. Okay. Round and green we got here. Okay. So next one, capital I, capital Y, round yellow. Okay. Round green again, students. Round green again. Round green. Small y, small y. Okay. Next one, round yellow. Okay. Round yellow. Okay. But if you see this one, students, it is going to be smaller. Smaller is wrinkled. Yes or no? Smaller is wrinkled. And here we capital Y. So can I say this is wrinkled yellow right now? Yes or no, students? Wrinkled yellow. Okay. For this, I will make a triangle here, students. Wrinkled yellow is a triangle. Okay. Wrinkled yellow. Wrinkled yellow is our next one. Now this one also wrinkled yellow. Yes or no, students? Wrinkled yellow. Okay. Next one, students, round yellow. This one is round yellow again. Round yellow. This one, round small y. So can I tell round green here? Round green, this one. Okay. This one is again wrinkled yellow. Wrinkled yellow, students. Wrinkled yellow. Last one, students. Last one is going to be, this is wrinkled green. So I'll make a star here. So this is a star. Let's say this is a star. Okay. You have a star. That is your wrinkled green. wrinkled green. Now students, I want all of you to count. Can all of you count and tell me how many do we have round yellow? Students, count. 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay. How many round green? 1, 2, 3. How many wrinkled yellow? 1, 2, 3. And how many wrinkled green? 1. So by this Punnett square method, students, by this Punnett square method, we can get this is nothing but, students, this is nothing but the phenotypic ratio of your dihybrid cross, where these two are technically the parental type and these two are the recombinant type. Recombinant, because see, round yellow, wrinkled green, round yellow and wrinkled green was present here also. Round yellow, wrinkled green. So these two are parental, but these two are because of recombination. Mixture, these are mixture basically. These are basically mix and match. That is round and green and wrinkled yellow. Okay, those are recombinant type, students. Now, students, now, do you want a trick here? Because sometimes by chance, if they ask you the genotypic ratio, do you want a trick? All of you students, do you want a trick? All of you in the chat, yes or no first? If you say no, I'll go for it. If you say yes sir, I need a trick, then I'll give you a trick. How to find the genotypic ratio? One trick? Okay, listen to me, students. Listen to me, all of you listen to me. Right. Now, what if I say two monohybrid cross? If you take two monohybrid cross and you cross them together, two monohybrid cross, if you cross them together, we get a dihybrid. Now, what do I mean by that? Now, students, can anyone in the chat tell me, even in my class tell me, students, even in my class tell me, what is the phenotypic ratio in monohybrid cross? See, we take two monohybrid and we cross them and we get the dihybrid. Okay, let's say in the case of phenotypic, we have 3:1 is the phenotypic ratio in monohybrid. Cross it with 3:1. What do we get, students? What do we get? 3 3s are 9. 1 into 3, 3. 1 into 3, 3. 1 into 1, 1. Done. This is your genotypic ratio, yes or no? This right here is a phenotypic ratio of your dihybrid. Now, let's say genotypic ratio is what? 1:2:1 for monohybrid. 1:2:1 for one more monohybrid. Let's multiply. 1 into 1, 1. 1 into 2, 2. 1 into 1, 1. 2 into 1, 2. 2 into 2, 4. 2 into 1, 2. Again, 1 into 1, 1. 1 into 2, 2. And 1 into 1 is 1. And here we go. This right here is the genotypic ratio, student. This right here is a genotypic ratio. This right here is the genotypic ratio of your dihybrid cross. Done. Done, students. Done. Crazy, no, students? Crazy, no? Yes or no? Crazy, right? With the help of the simple trick, you can tell the genotypic ratio of your dihybrid also. Dihybrid also, students. Done. This is what is given here also. Now, students, now we come to something called as, from your dihybrid cross, we generally get something called as law of independent assortment. From monohybrid, we got law of segregation as well as law of independent assortment. So we got law of segregation, law of dominance from monohybrid. Now from dihybrid, we get this law of independent assortment. Now what does it say? What does it say? It says that Mendel's law of independent assortment states that when two pair of traits. Now students, first of all tell me, what is one pair of trait? One pair of trait means two traits. Pair always means two. A couple or a pair. Couple of years. Couple means two always. A pair of shoe. A pair means two generally. So one pair is two. So what do I mean by two pairs? Two pairs means four. Yes or no? So one pair means two. Two pair means four. So when there are four traits together, right? When two pairs of traits are combined in a hybrid, For example, students, for example, if you go back here, right? If you go back here, capital R, capital Y, small y. Here we have round, wrinkled, yellow, green. We have round, one trait, wrinkled, second trait, yellow, third trait, and green, fourth trait here. These four traits are coming from two characters, that is seed shape and seed color. Seed shape and seed color. So law of independent assortment says that, right? Whenever there is two characters together, seed shape, seed color together, right? Segregation of one pair of character, that is segregation of seed shape, will be independent of segregation of seed color. So seed shape will go separately, seed color will go separately. They do not mix among each other, students. Okay. So here, if you go back to this hybrid right now, here in this hybrid, the seed shape will segregate, seed color can segregate separately. That is called as law of independent assortment. When a hybrid, two characters can go separately. That's all. That's all. Okay. The students, in examination, they will not ask you this definition. They'll ask. In examination, they will never ask you this. In examination, what they will ask students? In examination, they will ask you question like this. In examination, they'll ask you question like this. How do you solve now? How do you solve this question? Anyone? Here, they'll give you options also. They'll give you option like this. 100%, 50 to 56%, 18%, 9%. So when you get these questions, you should be able to solve them. Okay. Now, so let's read the question. It says that what is the expected percentage of F2? They're asking you what is the expected percentage of F2 with yellow inflated pod. So they're asking the percentage of yellow inflated pod in dihybrid cross experiment involving the pea plants with green color. Students, here we have green color and inflated pod. We are crossing with yellow color constricted pod. Students, doesn't this look like doesn't it look like round yellow crossing with wrinkled green? Yes or no, students? See, here I am using, in this case I am using right now the example given your NCERT for dihybrid cross. These are two different characters here, seed shape and seed color, from the 7 characters taken by Mendel or 14 traits taken by Mendel. Now, is it always seed shape and seed color? No. So they can change the question. They can change the question from seed shape to seed color to pod shape and pod color. All of you understanding? Here in our example of dihybrid, we have taken seed shape and seed color. But in examination, they can give, let's say, they can give you something like flower position and flower color. Yes or no? Or they can give you any other different pod shape and pod color. Yes or no? So they can give you different characters also. So here in this question, they have given you different characters here. So again, students, green color inflated. These two are dominant. Like how we had yellow and round in yellow. Okay. Yellow and constricted pod here, both are your recessive traits. Recessive trait. Like wrinkled and green. Similar to wrinkled and green. Same thing is present. So students, doesn't matter whatever character or trait we take. Will the dihybrid ratio change? Never. The dihybrid ratio will never change. It will be, especially in Mendelian. In non-Mendelian it can change, but in Mendelian it will never change. It will be always 9:3:3:1 only. So this is, both are what? Both are your parents. So this parent was always 9. This parent was 1. So here also, this is going to be 9 and this is going to be what? 1 only. whatever hybrid which we get in the middle, whatever hybrid which we get in the middle is going to be what? 3:3 only. So in your example, they have given you one hybrid, that is yellow from here, right? Yellow from here and inflated from here. So between these they have made a hybrid. So the number of this hybrid, students, yellow and inflated number will be 3 of them. Like how we had here, round yellow, wrinkled green. So we took round from here and green from here. These two we made round and green. Now then we took wrinkled from here and yellow from here. We made a hybrid here. These two are hybrids. Similarly, here also we can get a hybrid. So hybrid will be always 3. These two are parentals. Now once you have identified the hybrid right now, done. Work is done, students. All you need to do is right now, number of hybrid, 3. Total students, tell me, how many are the total progenies? All of you, can anyone tell me what is the number of total progenies here? 16, yes or no? Count, all of you, count. 1, 2, 3, 4, 4. 4 to the 4, 4 to the 4, 4, 16. 16, right? 9 plus 1, 10. 10 plus 3, 13. 13 plus 3, 16. Right? So, total progeny is 16. So, 3 divided by 16 into 100. That is for percentage. Students, you get the answer. You get the answer, students. You get the answer now. All of it is the answer. 3 divided by 16. Now, students, I'll change the question now. Tell me the percentage. Tell me the percentage of, let's say, green color inflated. What percentage of F2 will be green inflated? That I want you to do. Very simple. In that case, you will take green inflated is 9. 9 divided by 16 into 100 in that case. Yes or no? Now, let's say I'll change the question. To, let's say, tell me the percentage of yellow constricted. So in that case, what you will do? In that case, you will do 1 divided by 16 into 100. Yes or no, students? Yes or no? Very simple, students. Very simple it is. I hope all of you are able to solve the numericals now. Very simple, students. Now, this numerical has a variation also. Now, students, this is how the variation comes. Now, what they'll do sometimes is that they will not ask you percentage. Instead of asking percentage, they'll give you a number only. See this. In a cross between yellow round and wrinkled green. Now, they have used typical dihybrid examples. Now, a population of 800 individuals were found in F2. What would be the number of yellow wrinkled seeds? Now, students, first of all, tell me, all of you tell me, how many yellow wrinkled do we have? Yellow wrinkled, how many do we have? Do we have 3? Do we have 3? Or do we have 1? Or do we have 9? Yellow wrinkled, tell me, students. Yellow wrinkled, we have how much? All of you. Yellow wrinkled, students. Yellow wrinkled is 3. It's a hybrid, obviously, because wrinkled green and yellow round are the parents here. Yellow wrinkled is a hybrid. It is a recombinant. So 3 are going to be there. Yellow wrinkled is 3 now. 3. Now, what you need to do is, again, in this case, 3 divided by 16. But instead of doing into 100, you do what? Into the number given here. What is the number? 800. Into 800, and you will get your answer, students. You will get your answer. That's all. I hope all of you are able to understand the variation also. This is how the variation is going to be. This is how the variation is going to be for this question also. So if they ask you percentage, do into 100. If they do not ask you percentage, multiply the F2 progenies itself. You'll get the number. Students, how is the genetics now? Students, are you able to understand? Easy, no, students? It's easy. It's a matter of how you approach the entire subject. Okay. Now, students, with that being said, let's hear some sad story now. Let's hear a sad story, all of you. Students, all of you, quickly smash your like button, all of you. Quickly smash your like button, all of you. Done? Done. Now, students. Students, sometimes what happens, whatever hard work you do in life, right? Whatever hard work you do in life, you might not see the result on the go. What do I mean by that, students? What do I mean by that? Imagine you study 3 hours today, 4 hours today. You will not see the result in 1 week or 2 weeks. Whatever hard work you're doing right now with me, the result will be shown on the exam day or the result day. Exam result will be shown. So, but sometimes life is very unpredictable and you do not see the result until you die also. It does happen. And same thing happened to Mendel also, Mendel also, students. Because in the case of Mendel, what happened was whatever work he did throughout his life, the 7 long years of your hybrid experiments and everything, Mendel died without even knowing the significance of his work. Why? Very simple, student. Let's say Ravi, Adina, all of you, right? Let's say you discover a new frog. Let's say you discover a frog in Western Ghats. Now, what is the first thing you will, students? First thing you will do? You will take a photo. You'll go take your phone. You will open. You will take a photo. You will take a photo here. And after taking the photo, what do? You upload on Instagram, upload on Facebook, upload on Snapchat. You keep on uploading. So as soon as you upload the image on the internet, it goes viral and you get the recognition. Yes or no? But students, during Mendel time, did we have internet? Students, during Mendel time, did we have internet? No, we didn't have internet. So Mendel published his work in 1865, but it remained unrecognized till 1900s. Completely unrecognized, students. What are the reasons? Right? Work could not be widely publicized because there is lack of network. Factors were stated as stable and discrete units by Mendel, and Mendel's concept of alleles never blend, that is law of segregation, right, was not accepted by your contemporaries, that is your fellow people. Use of mathematics to explain biological phenomena was never accepted. No physical proof for existence of factors, that is experimentally genes and alleles were not identified. And students, first of all, you tell me, students, first of all, if I teach you maths in biology, you'll not like it. Obviously, you'll like, sir, why maths in biology? Now, same thing happened back then also. When he started to use mathematics in biology, he was not accepted by the entire population. That is what happened back then. Students. So, because of all of these reasons, Mendel's work was not published. Mendel's work did not get recognition, and Mendel died eventually, students. And all of you know, dead people cannot get Nobel Prize. Yes, sorry, students. Dead people cannot get Nobel Prize. But students, but all the sad story aside, do you think hard work goes to waste? All of you, do you think hard work will ever go to waste, students? Hard work will ever go to waste? No. Hard work never goes to waste whatsoever, because there is always light at the end of the tunnel. There's always light. That is what happened with Mendel, students. Mendel also. There were 3 different scientists who individually, when they were working on a project, they realized, wait a minute, there was a scientist before who was actually explained everything, and that is where 3 different scientists independently rediscovered the Mendel's work. And as we know right now, the genetics came into existence. The entire advancement in genetics is because of Mendel, and these 3 scientists rediscovered the Mendel's work. Hugo de Veris, Carl Collins, and Erich von Schmuck, right? Schmuck. 3 different scientists rediscovered the Mendel's work on inheritance of character, students. 3 different scientists. So hard work pays off. So all of you need to do hard work also, students. Hard work. Okay. Now, students. Now with that, we come to something called as chromosomal theory of inheritance. Why? Because after the rediscovering of Mendel's work, there was advancement in microscopy also. All microscopes, right, they were discovered properly. Yes. So after the discovery of microscope, there were 2 main scientists, along with even T.H. Morgan, right, called as Certain and Theodore Boveri, students. These 2 scientists, gave something called as chromosomal theory of inheritance, students. Shall we start, all of you? Students, shall we start? All of you, shall we start? Yes or no? Shall we start? Now, students, how many of you feel it's a difficult concept to understand? Raise your hands. How many of you feel this is very difficult to understand, sir? I cannot understand this. Raise your hands. Raise your hands. All of you, raise your hands. Students, raise your hands. How many of you feel this is a difficult concept to understand? Difficult concept to understand, students. Raise your hands, all of you. Raise your hands. So I'm not able to understand this. Okay. Let me tell you one very students. I need a volunteer. Students, I need a volunteer. Who wants to volunteer? I'll teach you this in a very simple way. I need a volunteer, students. I need a volunteer. Okay. Let's take today. I like Ravi name today. Ravi. We'll take Ravi because my brother's name is also Ravi. Okay. Okay. Let's take Ravi, students. Okay. Now let's say here we have Ravi here. Okay. Let's say we have Ravi. Okay. This is your Ravi here. Let's give Ravi some nice hair now. We'll give green hair to Ravi here. Full fancy. Ravi is very fancy. Okay. Now students, now this Ravi, let's say he has to go to his friend's house now. This is your friend. This is your friend's house here. Okay. Ravi has to go to the friend's house here. Okay. Now students, distance is around 10 kilometers. 10 kilometers, students. 10 kilometers. Let's say 10 kilometers too less. Distance is around 35 kilometers. Okay. 35 kilometers. Now, how do you think, students? How do you think Ravi will go right now, students? How do you think Ravi will go from here to here? Can he jog? Cycle? No. He'll take a bike or he'll take a car. Now, let's say Ravi has a lot of money here. Ravi is very rich person here, very rich person. All of you ask him some money, okay? Now, Ravi has a car. Let's say Ravi has a car right now, students. Ravi has a car here. Ravi has a bus only, students. Ravi has a TT only, let's say. Ravi has a TT only. Okay. So, Ravi is inside a car now or a bus. Ravi has a bus only, let's say. Very, very rich. Car is for local people. He has his own bus only. He has his own bus. Okay, we'll put light here. Light is coming out. Okay. Now, students, all of you tell me right now, students. Now, if this bus is going from here to here, will Ravi also move along with the bus? Yes or no? If this bus is going from here to here, will Ravi also move from here to here? Yes or no, students? Yes or no? All of you. If Ravi is going from here, if the bus is moving, Ravi will also move. Why? Because Ravi is inside the bus now. All of you students, Ravi is present where? Inside the bus. Yes or no? So if the bus is moving, Ravi will also move. Yes. So same concept also applies here, students. Now here, let's say this chromosome is here. Chromosome is like a bus. Chromosome is like a bus right now. Chromosome is like a bus. Okay. Chromosomes are like your bus. Now, and these genes, basically, these genes present here. There are multiple genes present, right? Genes and alleles here. Genes and alleles present here. These are genes. Genes are like your Ravi. Okay. Now, students, all of you tell me in the chat right now, if this chromosome separate like this, if this chromosomes go this side and if this chromosome moves this side, let's say the homologous chromosomes are separating in the anaphase. If the chromosomes are separating, will the alleles present here will also separate? See, bus is moving. If bus is moving, even Ravi is moving along with it because Ravi is present inside the bus. Now, if this chromosome is moving this side, this chromosome is moving this side, the alleles present on the chromosome, alleles also move. All of you agree with me? Just like how Ravi is moving along with the bus because Ravi is present inside the bus, here also alleles are present inside a chromosome. If this chromosome will move, alleles will also move. Simple logic. That's all is the concept. Students, that's all is the concept. That's all is chromosomal theory of inheritance. That is all is chromosomal theory of inheritance, students. That is, behavior of chromosome and behavior of a gene is parallel to each other. Behavior of a chromosome and behavior of a gene is parallel to each other. Because technically speaking, gene is present on the chromosome only. Okay. Easy or not? Tell me, students. Easy or not? Okay. Now, before this conclusion, lot of work was done actually. Right. Here we have your Water and Saturn worked on your lubber grasshopper. He noticed that chromosomes occur in pair. From his experiment, he observed that chromosomes occur in pair. And then we have Theodore Boveri. He worked on your Ascaris. He noticed that during meiosis, reduction division of chromosome in germ cells happens, right? There is a reduction in happening. That's all. Chromosomes segregate during gamete formation. All of you know this, right? Chromosomes segregate during gamete formation. He observed that. Then certain also observed that the chromosome pair of the homologous pair of chromosomes align themselves on metaphase 1 plate randomly and separation during anaphase 1. So all of you remember, during anaphase 1 the homologous chromosomes separating, meiosis. Okay, so these were the points given by chromosomal theory of inheritance. Chromosomes occur in pair, true. Chromosomes segregate during gamete formation, true. Chromosomes segregate independently of each other, that is, there are two chromosomes, one will go this side, one will go this side. They are segregating, right? Segregating. Now students, doesn't this look very similar to your Mendel work also? Because Mendel told us that factors occur in pair. We have capital T, small t. Yes, factors segregate during gamete formation. During gamete formation, capital T comes here, small t comes here, and factors segregate independent of each other. Law of independent assortment. So students, do all of you see a similarity between Mendel's work as well as your chromosomal theory of inheritance? Yes, it does. So why is there a similarity? There is a similarity because genes are present. Students, genes are present where? Tell me students, genes are present where? Genes are present on the chromosome. Behavior of a chromosome is similar to behavior of a gene. Okay, so genes are found on the specific location of the chromosome, and behavior of these chromosomes during meiosis can be explain the Mendel's law of inheritance. That's all students. That's all is the concept. That is all is the concept. Behavior of a gene is similar to behavior of chromosome. That's all students. That's all. Okay, they observed that behavior of a chromosome is parallel to the behavior of a gene. They used chromosome movement during cell division to explain Mendel's law of inheritance. So basically, Mendel's law of inheritance was explained very well using chromosomes. Why? Because genes are present on the chromosome. Students, how many of you are understanding? Raise your hands. How many of you are understanding the simple concept of chromosomal theory of inheritance? This is what is given your NCERT. Here you can see students, here we have G1, S, G2, S is missing here. Here we have S is missing here. See, anaphase 1, anaphase 2. In anaphase 1, these chromosomes separate here, this separate here. That is separation of here homologous chromosome separate here. This is anaphase 2. This is your anaphase 2. What is happening here? Sister chromatid is separating here. Sister chromatid is separating here. That's all students. Students, obviously I know Kannada students. I'm from Karnataka. I'm from Bangalore. Obviously I know Kannada baratte. Students, that is all. Students, can I get a quick answer? All of you know the concept right now. This is very easy right now, students. This is very easy right now. All of you tell the answer quickly. Students, all of you smash your like button. I want all of you to call your friends also, because everyone thinks this chapter is difficult, but it is not. It is not difficult chapter. It is a very easy chapter. I'll make it easy for you. In fact, I'll make every single chapter easy for you. If you follow me, I'll make every single chapter easy. It could be plant kingdom, animal kingdom. It is Gopika Ma'am's chapter. Every single chapter I'll make it easy, students. Okay. Now students, now we get the experimental proof. The experimental proof was given by whom? It was given by T.H. Morgan, students. T.H. Morgan. Then came the T.H. Morgan, students. T.H. Morgan. Thomas Hunt Morgan. He worked on Drosophila melanogaster. Students. Okay. Now students, is it easy to trap? Is it very easy to trap this? Very easy, students. Very easy. To trap fruit fly, all you need to do is take a banana peel, throw the banana peel in one corner, you will get fruit fly. You will get fruit flies, students. Fruit fly. Can you see? Fruit flies. So easy to harvest. So they can ask you questions. They can ask you question, why fruit fly? Student, very simple answer. Right. First point, they could be grown on the simple synthetic medium in the laboratory. Throw banana, you'll get it. Okay. They complete their life cycle in about 2 weeks, and a single mating could produce a large number of progenies. So you can unlimited supply also here. There was a clear differentiation of the sexes. Male and female can be seen very easily. Male is small, female is large here, right? Distribution feature. Also, it also has many types of hereditary variations that can be seen through low power microscope. That is body color, eye color, wing size, everything can be seen very easily. That is why Mendel chose it, students. This is very easy to remember, but students, how many of you do not understand linkage and recombination? Raise your hands. How many students? How many of you? How many of you do not understand linkage and recombination? Raise your hands, students. Raise your hands. All of you. Linkage and recombination. Because I will make this concept so easy for all of you that you will be able to teach your friends also. Students, I'll make this concept so easy for you, you will be able to teach your friends also, students. Friends also. Shall we start? Let me drink some water. And students, and after this concept right now, you will be able to tell me, you'll be able to tell me why Mendel was lucky. Students, why Mendel was lucky also. Students, if you think it is difficult, students, difficult to understand, shall I make it easy for all of you? Nandu, Prakash, yes, MS, Anish, I'll make this concept easy for you. Okay, shall we start? Bro, why are you looking for single people? Go study somewhere. Wow. Okay, so shall we start? Okay. I need 2 volunteers this time. I need 2 volunteers. I need two volunteers for this experiment. We'll do experiment now. I need 2 volunteers. Okay. Let's take Ravi student. Ravi is my favorite student now. Ravi is my favorite student. So let's take Ravi only. Okay. Let's take Ravi only right now. Okay. Now let's say we have Ravi here. Students, Ravi had which color hair? Students, Ravi had which color hair? Anyone? Ravi had which color hair? Anyone remember? Green, no? Okay. He had green color hair. Okay. Let's do Ravi here, students. But still, we have Ravi here. Okay. We need one more friend. Who is Ravi's friend here? Okay. Let's take MS. We have MS here. I can't read the whole name, sorry. Okay. This person will give him red color hair. Okay. Now we need one more person. We need Saif. We'll take Saif. Okay, no. We'll take Nandu. Nandu, Papa. Nandu is here. We'll take Nandu also. Nandu will give pink color hair. Okay, pink color hair will give. Students, pink is little chapri. We'll give blue color. Blue all same thing only. What to do? Okay. We'll give blue color hair. Okay, now students. Let's say here I have Ravi is here. MS is here. And we have Nandu here. Okay. Now students, this here I want all of you to understand. Ravi and MS both are BFF. Students, what is BFF? Students, what is BFF? All of you. It's fine. Now we will put regular. Tell me, students, what is BFF? BFF is best friends forever. Okay, they are what, students? They are best friends forever. Now students, if Ravi and MS are BFF, will they fight among each other, students? No. Will they have very good linkage among each other? Link is very nice. Their link is very nice. Their BFF. So linkage is very high. Yes or no, students? Okay. So can I tell between Ravi and MS, linkage is very high? If linkage is very high, do they fight? They don't fight. They don't fight only. Okay. So recombination is very low. Recombination is very low, students. Now, students, now we have the fight. Ravi and Nandu, like next level fighting is happening between them. He will bring one gun. He will bring one much, like full fight. Full fight is happening. Okay. Now between Nandu and Ravi, full cold war and fight is happening. Now, students, tell me, if Nandu and Ravi fight is happening between them, do they have linkage? No. They don't have linkage. They do not have linkage. So between Nandu and Ravi, which are very far from each other, students. First of all, they're very far from each other. Okay. So between them, linkage is not there. Will they fight? Obviously, students. Recombination is very high. Recombination is very high, students. Okay. Now, students, now let's take this concept into a chromosome right now. Now, let's say, students, here Ravi is gene A, MS is gene B, and Nandu is gene G. Now, if I draw chromosome here, okay, if I draw chromosome, students, here, I have a chromosome, students. This is a chromosome right now. Let's say this is a chromosome. On this chromosome, students, I have a chromosome here, students. Now, on this chromosome right now, let's say I have gene A here, that is your Ravi, gene B here, that is your MS, and let's say I have gene C here, that is your Nandu. The students, tell me, students, all of you tell me right now, all of you tell me, between your gene A and gene B, when two genes are tightly linked, that is, when two genes are very close to each other, will the linkage be high or low? All of you in the chat, tell me. When two genes are very close to each other on a chromosome, will the linkage be high or low? All of you in the chat, all of you. When two genes are very close to each other, their linkage is very high and recombination is very low. Very low. Two genes are very close to each other. Now let's say, let's take, if the two genes are very far from each other, in this case A and C, A and C, A and C, they are far from each other, they are far from each other. When two genes are very far from each other, yes or no? Far from each other, in this case, in that case, between gene A and gene C, they're loosely linked, so linkage is very low, recombination is very high. Recombination is very high, students. So can I tell right now, linkage is technically inversely proportionate to recombination? All of you can agree on this. If linkage is high, recombination is low. Yes or no, students? Yes or no? All of you. Now, let me show you one diagram which will make you better understanding, students. If you see, this is what is given in NCERT, students. This is what is given in NCERT. Let me open NCERT for that, students. Students, here what this is a table is given you. One is chromosome, one is gene here. They both are similarities basically. Okay. Now, students, if you see here, all of you, tell me, students, these two genes are very close to each other or far from each other, all of you? Y and W, students. These genes on this one chromosome itself, they're very what? Very close to each other. So linkage is high, recombination is low. So can you see, parental type is your linkage 98.7, very high. Recombination is very low here, 1.3%. 1.3% here, because linkage is very high here. Now, if you look at this one, students, if you look at this here, in this example, can you see, students, here? Two genes are very far from each other. Here, students, all of you, here, two genes are far from each other. Tell me, students, but they are present on the same chromosome only. They are present on the same chromosome only. So, can you see, automatically, linkage is coming down, and can you see, recombination is going up now. 37.2% recombination, because genes are far from each other. Okay. Now, students, what should happen? Now, let's say here, if you go back to this diagram right now, students. Now, let's say I have one gene on this chromosome. Similarly, students, I have one more chromosome here, and here I have one more gene here. One more gene here, students. These, let's say here we have gene D here, gene C is here, gene D is here. Okay. Now students, tell me, in this case, will the recombination be high or low? Tell me students, in this case, recombination will be high or low? See, two genes are very far from each other. If they are far from each other, students, linkage is low and recombination is going to be very high. Yes or students? Yes or no? Recombination is going to be very high in that case. Very high recombination. If you do a crossing over, with this chromosome and this chromosome, if there is a crossing over, by chance, it doesn't happen usually, in that case, very high it will become. Students, right now, you learnt a beautiful concept right now. That is linkage and recombination, students. Linkage and recombination concept. So, can you see here? They are linked, they are not linked here, and not linked again, very far from each other. Okay. So, that is linkage and recombination. Morgan observed that two genes, students, very important. Morgan observed that two genes did not segregate independently of each other. So, let's say, students, now let's say, all of you tell me in the chat right now. Let's say I have one chromosome here. Okay. Here I have one gene here for, let's say, seed shape, and one gene here for seed color. So tell me, students, in this case, the two genes are close or very far from each other? Tell me. These two genes are very close to each other. They're very close to each other, right? Very close to each other, students. In this case, linkage is going to be high. And students, do you think they can show recombination? Can they show recombination? Generally not. Because these two genes are very close to each other, recombination is generally not seen here. So, recombination is very low or not present. Now, students, all of you tell me, if this was the case, right, seed shape and seed color, if they're very close to each other, do you think we would get this ratio here? Is it possible to get this ratio? 3 and 3? Never. Because recombination is not happening only. If seed shape and seed color is so close to each other, we would not get this recombination at all. So, Mendel was very lucky. Because all the work which he did, all these different characters he took, traits he took, right? They were present on different chromosomes. Now, students, you tell me, what if Mendel took some plant, where genes were very close to each other? If the genes were very close to each other, would we get this ratio? Students, if Mendel did the experiment with some other plant, not pea plant, some other plant, okay, where genes were accidentally too close to each other, because he doesn't know where are genes present, because microscope is not there, hit and trial method, would he get the same result? Students, will Mendel get the same result if genes were very close to each other in some plant? Tell me, students, would Mendel get the same 9:3:3:1 ratio if he took a plant where genes were very close to each other? He would not get the same result, and that actually happened. Mendel worked on hawkweed, he worked on beans. When he worked on hawkweed and beans, Mendel could not get the same result. Students, that's why we say luck also is very important. Sometimes. So Mendel was lucky in the starting to take pea plant, and in the case of pea plant, the chromosomes were genes were present on different chromosomes, and he could get recombination easily. But when he worked on other plants, he did not get it. Mendel did not get it, students. Mendel did not get the same result. Okay, that is linkage and recombination. Beautiful, no, students? Beautiful, no? So the F2 ratio deviated. There was a change from 9:3:1 ratio. He could not get the same ratio. There was a different ratio when Morgan did the experiment. When Morgan did the experiment, he got different ratio only. Okay. So Morgan attributed this to physical association. That is, physical association. That is physical association of genes, gene A, gene B, gene C. Physical association of genes on a chromosome is called as linkage. It is called as what? Linkage. Okay. Use the term modern attribute the physical association of genes or linkage of these two genes. Okay. The term recombination for generation of non-parallel gene combination. Okay, that is called as recombination. Some genes are very tightly linked, show very low recombination. Tightly linked means very high linkage. Some genes are loosely linked, very far from each other. Linkage is low. Recombination is high here. Students, with that, that concept is also over. Now, students, there is one more scientist called as Alfred Stewart Went. Alfred Stewart Went is that, students. Alfred Stewart Went was the student of T.H. Morgan. He was a student of T.H. Morgan. Okay. He did something called as recombinant frequency. He did something, students. Morgan student Alfred Stewart mapped the gene's position on the chromosome. He used something called as frequency of recombination. That is how often the recombination is happening. That is what was studied by Alfred Stewart Went. So Alfred Stewart Went observed how often this recombination is happening, and based on this he made something called as gene map. He gave something called as gene map. In this gene map we can tell how far the genes are. With the help of this gene map we can tell the distance between two genes. This is like map units or centimorgan. So map unit or centimorgan is the unit here, or centimorgan. That is distance between two genes. What is the distance between this and this gene? This and this gene. That is called as map unit or centimorgan gene. That was done by Alfred Stewart Went. That was done by our Alfred Stewart Went. Okay, right. He used frequency of recombination between gene pairs on the same chromosome as a measure of distance between the genes. Okay, that is called as your map unit or centimorgan, students. Now here is a quick question, students. Can anyone tell the answer? Frequency of recombination between gene pair on the same chromosome as a measure of distance between the genes. That is your Alfred Stewart Went. This is a NEET PYQ, 2023 NEET PYQ. Okay, students, here is the question. Start solving, students. Start solving. Start solving, students. Start solving. I'll give you exactly 1 minute. Go, students, go. You'll get 1 minute to solve this question. It's an AR question. Properly read and answer. Okay. See, students, Mendel's law of independent assortment does not hold good for genes that are located close to each other on the same chromosome. Obviously, if the linkage is very high, Mendel's law is not proper. Yes, sir, students. Yes or no? That is, Mendel's law of independent assortment does not hold good for genes that are located close to each other. Okay. And reason, students? Closely located genes assort independent, students. If the two genes are close to each other, students, do you think they will go separate in recombination? Tell me, students, do they go separate in recombination? Or if the two genes are close to each other, will they go together, students? Will they go together? Students, I told you, if the two genes are very close to each other, the linkage is very high. If the linkage is very high, why will they assort independent, students? Why will they show independent assortment? They will not show independent assortment, students. They will never show independent assortment. That is, closely located genes assort independently. No, they will assort together. They will go together always. They will always go together. So what is the answer, students? A is true, R is false actually. This is false. They will never assort independently. That is, closely located genes will not go separate, they'll go together. Why will they assort independently, students? Why they have to assort independently? Okay. Here's the question, students. Here's the question. This I didn't teach you, so I'll tell the answer, students. In Drosophila, the genes for color of the body and everything were present on the X chromosome. All the work was done on the X chromosome, students. All the present was done on X chromosome. Go. Tell the answer, student. Tell the answer. You will get 30 seconds. You will get 30 seconds from my side. All these are NEET PYQs. All of these questions are NEET PYQs, student. NEET PYQs. Go. Welcome, welcome, welcome. Welcome, Ravi. Solve the question, students. Solve the question. Okay, question is very simple. So the question says low frequency recombination. That is, very low recombination is there. Okay. Very low frequency of recombination is there. If low recombination means high linkage. What is the meaning of high linkage, students? What is the meaning of high linkage? All of you. The meaning of high linkage is what? Two genes are close to each other. All of you, yes or no? If two genes are very close to each other, then linkage is very high. Yes or no? Linkage is very high. Okay, students. Now, so with that, we come to the end of linkage and recombination. With that, we come to the end of linkage and recombination. Now, students, I'll be teaching you two most important topics right now, which has been repeated in examination multiple times, called as polygenetic inheritance and pleiotropy. Okay, students, how many of you find it difficult? Raise your hands. How many of you find it difficult to understand pleiotropy as well as the polygenetic inheritance? Difficult to understand? All of you. Raise your hands. Students, raise your hands. How many of you find it difficult to understand pleiotropy as well as polygenetic inheritance? Raise your hands. Because I want to make it very easy for all of you right now. Okay, students. Okay. Now, according to Mendel sir, right? According to Mendel sir, we will always have tall plants and dwarf plants. Yes or no? According to Mendel, we will always have a tall plant and dwarf plant. But is it possible to have intermediate in the case of Mendel? Do we find medium height in Mendel's work? Students, tell me yes or no. Students, tell me yes or no. According to Mendel, we have your green or yellow, round or wrinkled, full, constricted. Yes or no? So according to Mendel, we always have something called as your two separate ends, tall, short. There is never intermediate. But if you look at the humans, our humans, do we have gradation in height? Do we have 5'5", 5'6", 5'7", 5'8", 5'9", 5'10", 5'11", 6 feet, 6'1", 6'2", 6'3", 6'4"? All of you. So in the case of Mendel, did we have a gradation in the case of height? Yes, we do. Okay, what about human skin color, students? Human skin color. In human skin color also, we have a gradation, right? We do not have full black, full white. We have brown skin color. Yes, sir, students. We have the brown skin color. We have light brown, dark brown. Yes, sir, students. So all of these concepts where we see a gradation, that is explained by polygenetic inheritance. That is explained by polygenetic inheritance. So as you can see, we have different emojis. Yes or no? We have different emojis for every single skin color. Every single skin color, we have different emoji. All of this can be observed. All of this can be explained with the help of polygenetic inheritance, students. Polygenetic inheritance. Okay. Now, what is the meaning of poly, students? Tell me, poly means one or many. All of it, tell me, students. What is the meaning of poly? Poly means one or many, students. Tell me. Anyone in the chat? Poly means one or many. All of you. See, poly means many. Genic means genes. So in polygenetic inheritance, we have many genes controlling one trait. Many genes controlling one trait. Many genes are controlling one trait. Okay. And students, this one trait is here, right? This one trait, right? One trait can also be environmental influenced also, and this one trait will show something called as cumulative effect. What do I mean by that? Now let's take an example. Let's take an example of human skin color. the human skin color will change from person to person. Why? Because of a pigment. Anyone knows which pigment, students? Which pigment? This dark person will have a lot of that pigment, while this light skin person will have very less pigment. The pigment is called as melanin pigment. It is called as melanin pigment. And this entire melanin pigment is controlled by three different genes. Three different genes. Okay. Melanin, not melatonin. Melatonin is different. Melanin. Okay. Let's say this entire melanin is controlled by three genes. Okay. We have gene A, gene B, and gene C. Three different genes are present. Now, students, what do I mean by cumulative or additive effect? The meaning of cumulative as well as additive effect is that whenever we have all dominant alleles, the trait is expressed very high trait. So this person will have very dark skin color. Okay, dark skin color. Okay. Now let's say small a. There are no dominant alleles only, no dominant alleles in this light skin color. Light skin color, students. Now what about we have capital A, capital B, small b, capital C, small c. In this we will have brown skin color. Okay. Now let's say small a, capital B, small b, small c. In this case, light brown color. Light brown is seen, students. So do you notice this is called as your cumulative effect. It is called as cumulative effect, where more dominant alleles are present, stronger is the expression. Stronger is expression right now. And it is also environmentally influenced. Students, what do I mean by that? Okay. Let's say this person here. If I take this person here and I keep it in sunlight 24/7 throughout the year, not 24/7, every single day 4 to 5 hours in sunlight for throughout the year, will the skin color change, students? Yes or no? There will be something called tanning. Tanning will happen, tanning. Yes or no? Tanning will happen, tan. So environmentally influenced. Same goes for human height also. In the case of human height, what happens, students? Human height, if you do not get proper nutrition, if you do not get proper vitamins, proper minerals, the height cannot grow sometimes. You might have, students, you might have very good genetics, but if you do not have proper nutrition, sometimes you might not grow tall also. Because genetics plays a role, but also it is environmentally influenced. Human height and human skin color. Okay, human height as well as human skin color, it is environmentally influenced. It is also environmentally influenced also. Environmentally influenced. Okay, students, done. Positive inheritance. Now, students, your pleiotropy is completely opposite of it. Pleiotropy is completely opposite, because in pleiotropy we have not many genes. One gene is present here. One gene can show multiple effects. Multiple effects, students. Effects. One gene can show multiple effects. Now you might ask me, sir, how is it possible? Students, how is it possible? Because this one gene can take part in the metabolic pathways. It can alter the metabolic pathways and it can show multiple effects. Multiple effects. Okay. So one gene here, students. One gene, multiple effects, students. It is, students, the best way to remember is by like a mother. Mother will do everything. She'll cook for you, she'll clean for you, she'll do everything for you. Similarly, like mother. This is like a pleiotropy is like your mother. Okay. Pleo means generally more. Pleo means generally more. Okay. More effects are seen by one gene. More effects is seen by one gene, students. One gene here. Okay. Now here, example, students. Example is phenylketonuria is an example here. Example, students, is here. Phenylketonuria is an example, students. This disease I'll teach you in the end, students. This disease I'll teach you in the end. Why is it caused? Because of there is an enzyme is not present. Phenylalanine hydrolase. The enzyme is not present. Because of the lack of enzyme, there are multiple variations that I'll teach you in the end. Don't worry. Now, students, can you see? Because of one gene is getting affected here. One gene is getting affected here. It can show multiple effects. Multiple effects. Mental retardedness, hair loss, skin pigmentation can be seen. So one gene, multiple effects. Multiple effects, students, multiple effects. Okay. Students, here is a question for all of you. Students, here's the question for all of you. Students, quickly smash your like button, all of you. Students, quickly smash your like button, all of you. Yes or no? We have to make 100 likes. We have to make 100 likes quickly. And students, I see many students are dropping out already, dropping out. Pleiotropy. Answer. The phenomena of pleiotropism refers to more than two genes affecting, no, one gene is present. Several alleles of a gene control single crossover, no. Presence of two alleles each of the two genes, no. That is a single gene control affecting multiple phenotypic expression. One gene, multiple expression, students. One gene, multiple expression. Start solving. Select the correct statements with respect to the pleiotropism. Pleiotropism, students. A gene is said to be pleiotropy if it affects more than one trait. Multiple traits can be affected, students. Okay, multiple traits can be affected here. Okay, pleiotropism is an example of pleiotropy. Yes, a condition where one gene has several alleles, it is referred to as pleiotropism. No, that is multiple allelism. This is multiple allelism. Okay, a trait is said to be pleiotropic. Several genes can be, no. That is polygenetic inheritance. This is polygenetic inheritance. Okay. So what is the answer, students? A and B only. A and B only. Okay. Students, with that, we come to the next topic called as sex determination, students. Shall we start? Students, do you realize we are finishing topic after topic? Topic after topic is getting over one by one. We are finished with the pleiotropy as well as your polygenetic inheritance. We finished the chromosomal theory of inheritance. We finished the dihybrid cross. We finished the star synthesis. So similarly, now you are left with sex determination, mutation, pedigree and diseases. This will get over in 10 to 15 minutes. Mutation 5 to 10 minutes. Pedigree analysis 10 minutes. And entire disorders 15 minutes you'll get over. So roughly around, let's say around 45 minutes we can wrap up the chapter. Okay, or if we want, if 45 to 1 hour we can wrap up the chapter 45 minutes. Technically 45 minutes is enough, right? So shall we start? Shall we start? Next topic called as sex determination. All of you students, there is no energy from all of you. Students, there is no energy only from all of you. Tri-hybrid cross is not there in syllabus. Tri-hybrid cross is not there. Okay. So where is the energy, all of you? All of you sleeping or what already? Huh? What is this? You are getting top level content for free of cost and still all of you are not showing energy on the top. Students, trust me, this top level content you will not find anywhere. Very easily you will get many teachers who will teach you such easy way. Such easily I am teaching you every single concept. Because everything is easy only, first of all. Okay. Students, shall we start next topic? Okay. Now students, the next topic is very simple. Okay. It is called as sex determination. All of you might be aware of it. Now students, I want all of you to understand one small fact here. Sex determination is generally genotypic based on which chromosome is entering, but also in some cases the sex determination is also environmentally influenced. Students, how many of you knew this? Students, how many of you knew this, that sex determination can happen because of environment also? I'll give you an example. In the case of turtle, students, in the case of turtle, let's have an egg. In the case of turtle, let's say I have an egg here. Now, if this egg is incubated, let's say very low temperature, then this egg will become male. Let's say there is very high temperature, incubation, very high temperature, this can become female. Students, how many of you knew that? Students, how many of you knew that? Students, how many of you knew that? This particular, right? This particular egg, low temperature, male. High temperature, it become female. That is based on environment. It is based on environment. Students, similarly is seen in the case of clownfish also, students. In the case of your clownfish also happens. In the case of clownfish, what happens is that this male is there, right? See, top is always female. If the top female dies, the next large male will convert the sex into a female. The male will become female there. Okay, that is seen in the case of clownfish also. Exchange. But we are not interested in that right now. We are interested in the normal sex determination that is based on chromosome. Okay. Now, students, now when I say right, when I say Finding Nemo, yes, Finding Nemo. Okay. So students, so technically speaking, in that Finding Nemo, right, that Nemo was there, dad was there, no dad. That dad can become mom also technically. Yes, sir, students. In Finding Nemo, the dad can become mom also there. Convert, because if the dad is larger there, the dad can get converted to mom there, technically speaking. Right. That's students. Now, in when I say evolution, which scientist comes to your mind? Evolution. Charles Darwin. When I say genetics, who comes to your mind? Genetics is Graham Mendel. When I say experimental genetics, T.H. Morgan. Yes or no? Okay. So when I say sex determination, students, sex determination, the scientist which needs to come to your mind is Henking. Henking is a scientist, students. Henking is a scientist. In the year 1891, he did crazy work. He did, students. He did crazy work. Okay. He worked on different animals, and he observed the spermatogenesis, students, spermatogenesis. And he observed that whenever there was spermatogenesis, he noticed that, students, 50% of sperms had something called as Y chromosome, and 50% of sperm did not have the Y chromosome. The Y chromosome was missing here. The Y chromosome was missing in 50% here. So that gave him an idea, students. That gave him an idea. So he told that in 50% of sperm, the Y chromosome is missing. In that case, he observed something called as X body. He observed X body. This is what he observed. In rest 50%, he observed the X body, students. X body. He observed the X body right now. X body. Okay, X body. And he later on, this X body right now is called as X chromosome. X chromosome. So initial discovery was done by your Henking itself. Students, with that, we come to the sex determination. Now here I'll be teaching you something called as male heterogamety as well as female heterogamety. Okay, students. Now what is male heterogamety? Very simple. Okay, so you understand the words first of all. Understand the words. It says male. Students, what is the meaning of hetero? Heterogamety. Heterogamety, students. Hetero means what? Hetero means different. Gamety means gamete. So in the case of male heterogamety, right, there are two different examples which you need to know. Okay, two different examples. The first example is in the case of human. The second example in the case of grasshopper. In both of these examples, right, generally speaking, right, here we have male and female. Here also we have male and female. Yes or no? Now, in the case of male heterogamety, it is always the males. It is always the males which have two different types of chromosomes, while female have only XX here. Same. In the case of grasshopper also, the female is XX here, while the male is X0. Okay. So the whole point is that, okay, the whole point is that in the case of male heterogamety, it is always the males. Okay, it is always the males which have, students, it is always the males which have the two different types of gametes. Here also males have two different types of gametes, students. Okay, students. All of you clear? Students, clear to all of you? Yes or no? Male heterogamity done? All of you. Male heterogamety done? Human and grasshopper. Male heterogamety. Okay. Now, students, now let's understand female heterogamety. Female heterogamety, students. Now, again, students, in female heterogamety, we have two different examples. Which color? Yellow, white one, two different examples. One is birds, one is butterfly. Okay, now let's draw male and female here. Male and female, male and female. Okay, student, once again, let me draw it properly only, otherwise it gets confused. Students, obviously, in the case of birds, the male will have ZZ and the female will have the ZW here. ZW, students, ZW. Okay. Now again here we will have ZZ here and Z naught here. So that is the female, student. That is the female heterogamety. The only difference is that in the case of female heterogamety, the females have two different type of gametes here. So whenever there are two different gametes, they can determine the sex of the offspring. Here, if the child has to be male or female, is decided by this male here. If the child has to be male or female, it is decided by here male here. If the child has to be male or female, decided by the female here. If the child has to be male or female, decided by the female here, students. That is called as male heterogamety and female heterogamety, students. But there is a special type of sex determination now. Now, this is mainly seen in the case of ants, certain type of your wasp and bees. Okay, in the case of bees generally, it is called as, students, haplodiploidy. Haplodiploidy sex determination, students. Now, students, this is very, very simple. Please understand what exactly is happening here. It is very, very easy, very easy. Okay. Now, let's say I have male here. Male is always haploid here. And we have female here. Male and female B. Female is always diploid, students. Female is always diploid here. Okay. Students, remember here 2n is equal to 32. So, here we have 16 and we have 32 chromosomes here. Now, male will produce, let's say, the sperm here, the gamete here, and the female will produce the egg here. Now, this is also haploid. This is also haploid, students. Now, all of you tell me in the chat right now, what needs to happen here? Diploid to haploid, always meiosis. Haploid to haploid, mitosis. Now, shocking. All of you, shocking. Whenever the sperm and egg fuse, it will always, it will always result in female only. And this is going to be diploid again. Diploid. Haploid plus haploid, diploid. And this female can later become queen or worker. Now, this queen or worker, students, do you know that? How can it become queen? How can it become vector? This entire classification is based on what food they get. Let's say the queen, let's say this female gets royal nectar. If this female gets the royal nectar, then queen. If there's no royal nectar, this queen will become worker. So this type of transformation is done based on the food which they get. Crazy, animal kingdom is, sorry, your entire insects are crazy, insects are crazy, Very crazy they are. Okay. Now, all of you might ask me, right? Sir, here we got female as a result of fertilization. Then how do we get male? Where are the male students? How are the males produced here? Students, very simple, very simple. Males are actually, students, this egg is here, right? This egg will directly develop into a male. And this is haploid. Males are always haploid. And this is possible because of a process called as, students, process called as what? Parthenogenesis. By the process called as what? Parthenogenesis. Because of parthenogenesis, this egg can directly develop into a male here. It can directly develop into the male right now. Okay. Now, students, now. Okay, let me just write it here separately. I need more space. Male haploid. Now, I want all of you to understand in this current scenario, in this, students, Sanjay, yes? Okay, students. Now, students, tell me, this male, do you think they have a father here? No. This male is always has a mother here. So, can I tell there is no father here? Father is absent. Father is absent, students. Father is absent here. Okay. Now, students, here, this male here, can he have a son? No. Always a female only, no. So, there is no son. No son. So, there is no father, there is no son. But, students, eventually speaking, this female will give rise to a male eventually by parthenogenesis. Yes. Now, I want all of you to understand. This female, like here, how we gave birth to this male, this female can give birth to a male. Yes. Now, this male does not have a father, but this female is developing from a male here. So, no father, but a grandfather here. There is a grandfather. There is a grandfather here. This male does not have a father, but have a grandfather. Now, this male here does not have a son, but has a grandson. Yes. So, there can be a grandson here, students. Grandson here. So, that is what is happening here, students. That is what is happening here. Okay. There is no father, there is no son, but there is a grandfather and there is a grandson. That is called as haplodiploidy types of sex determination, students. Sex determination. Done? Yes. Students, quickly smash your like button, all of you. Quick, very simple dimple it is. Very, very simple. That is sex determination. Done, all of you? Done? Understood? Students, if you understood, add some green hearts in the chat. Add some green hearts in the chat, students. Green hearts in the chat if you understood. All of you green hearts in the chat because I want to know if you're understanding. Because green is botany, no? Students, no. Here is a question for all of students. All of you. Question for all of you. Students, start solving, all of you. Question for all of you. That is, in honeybee population, sons do not have father but have grandfather. Right? They have grandfather. In haploidy in drones of honeybees due to the parthenogenesis, while fertilization is result of female. That is why we do not have father, right? Because of haplodiploidy. Because of parthenogenesis, it does not have a father. It doesn't have a father. Yes or no, students? It doesn't have a father. Okay. Okay. Darshini is coming now. At least learn from here. At least learn from here now. Okay. Now, students, now. Students, answer is shown to all of you, students. Answer is shown to all of you. Sad. Answer is shown by mistake. Okay, students. Solve this question. XO type of sex determination can be found in XO. XO type of sex determination can be found where? Students, which chapter next you want? From class 12th, which is the next chapter should we do? 12th, which one should we do? Next chapter from class 12th. Biotech. No, biotech ma'am will take, students. Biotech ma'am will take. Biotech I've thought before anyway. Biotech you can check, students. I am thinking finish microbes very easily. Yes, or no, students? We'll finish microbes. All of you. Will we finish microbes in the next class? All of you. Shall we finish microbes in the next class? Okay. Easy. Okay, students. Now we come to mutation. The next topic is called as mutation. Shall we start, students? Okay. All of you. Students, shall we start? Human health ma'am will teach, students. Human health ma'am will teach. Human health ma'am will teach. Ma'am is a pro there. Kovika ma'am is a pro who will teach you human health chapter. It's very like a story. Full story is there. Okay. But we will start with microbes. After microbes, I'll teach you something called as that's all. My botany is we have microbes, and that's all. Then you have ecology. What is this? There are no chapters for me in class 11th, class 12th. Okay, students. After this microbes, I'll teach you the entire part, remaining part, the entire remaining part. Okay, students. Mutation is what? Okay, students. What is mutation? Mutation is basically change in the genome. Change in the DNA is called as mutation. That is, the entire genetic genome is undergoing modification and it undergoes changes, and there is a change there. That is what is happening in the case of mutation. In the case of mutation, okay, that is what is happening in the case of mutation. No, Ravi was very serious today. That's why Ravi is my favorite student today. Darshini, you were before. Darshini, you are being replaced by Ravi now. Competition is very high. Okay. Now in mutation, students, it is also called as sudden inheritable change. It is a sudden inheritable change. Sudden inheritable change is called as, students, mutation. Inheritable change. Now, students, I want all of you to understand there are multiple types of mutation. Okay, there are multiple types of mutation, students. Multiple types of mutation. Okay. Now, there is something called as point mutation. Yes, so students, you might have learned about this point mutation before. In point mutation, what is happening? In the case of point mutation, here one nucleotide, every single, let's say this is your base here, right? One single base pair here will undergo, let's say, deleted here. Change is happening. Or this entire T, let's say, is replaced by cytosine. That is called as, your student, that is called as point mutation. That is called as point mutation right now. One single base pair is changing, that is point mutation. Then there is something called as frame shift mutation. Students, what is a frame shift mutation? Understand. In frame shift mutation, the entire reading frame is there, right? The entire reading frame undergoes changes. Why? Because if you remove one base pair or if you add one base pair, there will be complete change in the genome here. Okay. So students, as you can see here, let's say here we have thymine here. Now, if I completely remove this thymine, students, if I completely remove this thymine here, what will happen? This entire frame will go front right now. Yes or no, students? It will go front. See, here we had TAA was there, CTG. Now it is read by what? It is read by what? It is read by TA. This is constant. We have CGC. Now we have CGC here. That is, one thymine has been removed right now. So entire frame shift what happened. Okay. Now if you look at, this is your deletion. Now if you insert also, what happens? If you insert, now here, let's say you insert one nitrogenous base here. So entire frame shift goes this side right now. Okay. So we have CCT. Here we have CTG. So entire reading frame changes full. That is called as frame shift mutation. Frame shift mutation. Okay. Now, students, here I have to teach you one small thing. Please understand. Right. I'll write and teach you. There are something called as aneuploidy, euploidy, and everything. So please understand this and please write down also this one. Okay. Haryana se kon hai? I don't think anyone from Haryana hear us. We have cash army and bot army here. Okay. Now, students, now. I want all of you to understand there is something called as chromosomal changes. Now, all of these chromosomal changes are because of, Let's say there are homologous chromosomes don't separate out. There is no cytokinesis. That can lead to these conditions. Okay. Now, in chromosomal changes, broadly speaking, we have two categories. Okay. From top. Broadly speaking, we have two categories here. Two categories, students, two categories. One is called as aneuploidy. One is called as euploidy. Okay. Students, understand, students. This euploidy is mainly because of no cytokinesis generally. Whenever there is no cytokinesis, that can lead to euploidy. Okay. I saw Darshan I saw euploidy. In euploidy, what happens? There is a change in entire set. Change in set of chromosome. Set of chromosome. Let's say we had n becomes 2n, 2n becomes 3n, 3n becomes 4n. That is, we can find multi-nucleate condition can be seen here. Okay, the change in entire set, that is called as euploidy. That is called as euploidy. Almost well. That is called euploidy. Now, under aneuploidy, we have two types. One is called as your hypoploidy. One is called as hyperploidy. Students, understand what happens. In the case of hypoploidy, what happens, students? There is a decrease in number of chromosome. Hypoploidy. Chromosome number decreases. In hyperploidy, chromosome number increases here. Now, example you might understand, students. Decrease number of chromosomes, all of you know, right? There is something called as Turner syndrome. Turner syndrome, that is XO condition, which I'll teach you eventually. In your hyperploidy, students, we have your trisomy 21 Down syndrome, as well as Klinefelter syndrome. Students. So hyperploidy, increase number of chromosomes. Hypoploidy, decrease number of chromosomes. That is under aneuploidy. Euploidy, what is euploidy, students? Euploidy is what? Euploidy is there is no cytokinesis. Because there is no cytokinesis, what happens? Change in set of chromosome happens. Entire set changes. Okay, students. Easy, no? Easy, right? Thank you, students. Thank you. Huh? I'm also your fan, all of you, because you're sitting through my long lectures. All of you are sitting through my long lectures because I'm also your fan now. Okay, that is your end of mutation, students. Now, this entire mutation can be caused by multiple factors. It can be caused by your physical factors like your UV radiation, X-ray radiation. It can be caused because of chemicals, which people who smoke, smoking people and all, chemicals also. Okay, so there are multiple reasons. There are multiple mutation can happen. Okay, mutation can happen, students. Okay, now students, now we come to pedigree, students. Shall we start? I'll take only 5 minutes because here there is nothing to teach because I will give you extra points here. If you learn these points, every single pedigree can be easily be solved. I will give you a lot of hacks here. Ready, students? Ready? All of you ready, students, for pedigree? Students, all of you ready for pedigree or not? Ready or not, students? Quickly smash your like button. All of you quickly smash that like button right now. Smash that like button, students. All of you. Yes, I know faithful. Darshini, I know you're faithful, very. Students, who are my regular students? Can we see all the regular students in the class, in the chat? Who are my students? Who are my regular students? Regular students. See, I know Darshini, I know Jeannie, Adina is very regular, Prakash is very regular. Ravi, first time I'm seeing, students? No? Okay, students. Now, when you say pedigree, what comes to your mind, students? When you say pedigree, what comes to your mind? Dog biscuit, huh? Students, tell me. What, when I say pedigree, what comes to your mind? Dog biscuit? All of you. Yes or no? Tell me, students, tell me. Dog biscuit comes to your mind. Dog biscuit. Yes. Dog biscuit comes to your mind. It is not dog biscuit, students. Dog biscuit. And the whole meaning of pedigree, it originated from a. Let me show you, students. Let me show you. It originated from the French phrase, right? That is pied de. Students, I can't say French, obviously, right? It says pied de grue, which means foot of a crane. Like how a foot of a crane undergoes splitting like this, similarly a pedigree also undergoes splitting. Okay, the lines here are the split lines used in the chart, resembling the thin legs and a tiny foot of a crane. This is where the pedigree came into existence, word came into existence. Okay, but what is pedigree, student? Pedigree is basically analysis of traits in a several generations of a family is called as pedigree analysis. Let's say there's a disease in one family. The disease can be easily traced out in a family. Tracing can be done. Okay, tracing can be done with the help of pedigree analysis. Tracing can be done. Okay, inheritance of a particular trait is represented in the family tree over generation. In human genetics, it proves a strong tool for help in tracing the inheritance of a particular trait, abnormality, or disease. Let's say my great grandfather, my great grandfather had blue eyes, so I can also have blue eyes. Yes, I can also have blue eyes. Yes, sir, students, I can also have blue eyes. So that blue eyes trait has been passed down. It has been passed on from generation to generation. Blue eyes, hypnotize. Okay. Now, students, now here are all the symbols, students. Here are all the symbols. Now, students, generally in examination, they will ask you directly the symbol only. Symbol only they'll ask you, students. Symbol only. If they do not ask students, especially this one here, especially this one, the consanguineous mating one, they'll ask you the symbols directly they'll ask you in examination. Students, I'll give you 30 seconds. All of you stare at the symbol now. Stare. All of you stare at the symbol. 30 seconds. Time starts now, students. Time starts now. Go. Stare at the symbol, all of you. Nicely stare at it. Students, quickly smash your like button. I have to make 100 likes. 100 likes before the session ends. All of you. Okay. Now, students. Done? Okay. Now, students, let me show you a hack right now. Here is a hack. Okay, students. So I hope it is visible. If it is not visible, do not worry. You will get these notes in the Telegram channel, that is Vedantu NEET English Telegram channel, and also at my Telegram channel, that is Basavaraj Sir Biology. If you read these notes, if you read this entire thing, trust me, you can solve every single pedigree. Okay, it may not be clear to you right now, but once you get the notes, in the notes it will be very crystal clear to all of you. Crystal clear. Here it says autosomal dominant, skip generations, never. Affected individuals, both are equally affected here. Father to son is possible. All the special easy identification clues are given here. Clues. How to identify this disease. Let's say I'll just read for you. Read for you right now. Let's say for X-linked, what happens? In X-linked, what happens, students? X-linked. More males are affected here. Affected males get it from the mother. That is, from mother to son it goes generally. And right, mother to son it goes. Father will never give to son. From father to son it will never go. Daughter usually are carriers here. Unaffected parents can affect son. So if you read these clues, you can solve every single pedigree, students. Every single pedigree. Everything is given here, students. So you will take these notes and you can solve, students. You can start solving pedigree. Once you solve some pedigrees, you'll get a better idea. Okay. Now, students, there are two pedigree given your NCERT. One is your autosomal dominant. One is autosomal recessive. Autosomal dominant, students, example is what? The only example of autosomal dominant is myotonic dystrophy. The only example is called as myotonic dystrophy. You can see here, students. Myotonic dystrophy is given here. And this is not written anywhere in NCERT. It is written only here right now. Myotonic dystrophy written here. Autosomal dominant. Okay. This is autosomal recessive, that is sickle cell anemia. So try to identify, students. Just see the diagrams. Because in examination, they'll give you directly this one. Directly they'll give you. Okay, directly they'll give you, students. So students, solve this question. All of you students. Solve this question quickly. All of you solve. Yes or no? What is the answer? Which of the following represents mating between relatives or consanguineous mating? Which of the following represents consanguineous mating, students? Consanguineous mating. All of you. What is the answer? Option number 3. Students, Which of the following occurs due to presence of autosomal linked dominant trait? Autosomal linked dominant trait. MS, tell me the answer. Autosomal dominant trait. Myelin dystrophy. Okay. Study the pedigree chart given below. Identify the disorder. They can ask you a question like this. They can give you the diagram of NCR directly and they can ask you, identify the disease. Identify. Identify, students. Identify, all of you. All of you. That is your myelin dystrophy, autosomal dominant. Okay. Students, that is the end of pedigree also. Now we come to the final stretch. If you can give me next 15 to 20 minutes, 20 minutes, I'll finish the entire chapter. Students, should I finish the chapter now only or should we do part 3? Also tell me, students. Should we finish the chapter now only with genetic disorder or should we do a part 3? I don't think so. It makes sense. I don't think so. It makes sense to do a separate part for the only disorders. Yes or no? Next 20 to 25 minutes, maximum 25, we'll finish it off. Shall we start? Students, shall we start? I know all of you are sleepy, hungry, frustrated. Everything is there, but okay. But okay, students. We'll finish chapter today only. Sleepy also, I know. I understand it's already 10 o'clock, but we'll finish now only. We'll finish it today only. Now only, chapter should be Kel Katam. We say no, Kel Katam. Okay. हाँ मैं डेली लेता हूँ ऑब्वियसली लेता हूँ डेली नहीं लेता बट alternate days लेता okay shall we start okay students tomorrow I have a class at 6 o'clock okay now students genetic disorder now I'll give you a table students I'll give you a table okay the entire genetic disorders can be broadly classified into it can be broadly classified into students. Two categories. Okay. One is called as Mendelian disease. All of you write down. The first one is called as Mendelian disorder. Other one is called as chromosomal disorder. Okay. Now, under Mendelian disorder, students, all of you remember it is mainly because of alternation in a single gene generally. Okay. Now, under Mendelian disorder, students, under Mendelian disorder, we have two categories. One is called as autosomal. Okay, autosomal students. One is called as sex-linked. Okay. Now, students, under autosomal, we have two categories. Okay. Autosomal dominant. All of you, we have autosomal dominant, then we have autosomal recessive. Autosomal recessive, students. Autosomal dominant, autosomal recessive. Now, students, please write down. Under your autosomal dominant, there is only one disease called as MD in your NCRT. MD is nothing but students. MD is nothing but myotonic. Dystrophy. Myotonic dystrophy. Only one example. Dystrophy. Now, autosomal recessive, we have three diseases. One is PKU, phenylketonuria disease. One is your sickle cell anemia, that is sickle cell anemia. Third one is called as thalassemia. It's called as thalassemia, students. Three diseases. Under your autosomal recessive, we have thalassemia, students. Myotonic dystrophy, two of them, one of them, autosomal dominant, autosomal recessive, PKU, sickle cell anemia, thalassemia. Now, under sex linked, we have the color blindness. Then we have the entire hemophilia. Okay. Now, under your chromosomal also we have autosomal or sex linked. Under autosomal we have the Down syndrome. Under sex linked there is Turner syndrome as well as Klinefelter syndrome. And I'm going to move aside, students, I'm going to move aside, and all of you need to absorb this information. All of you absorb. PKU is phenylketonuria disease. Phenylketonuria disease, students. All of you. Notes, telegram. Phenylketonuria disease, students. Now, if you know this, you can solve so many questions. Let me give you one demo. How question will be asked generally in examination, students. See, question can be asked like this. In which disorder change of single base pair beta globin? No, this is different, students. Okay. Students, solve this question. Students, solve this question, all of you. See this. Which of the following is not a autosomal recessive disorder? See, we know PKU, sickle cell anemia, right? As for students, PKU, sickle cell anemia, as well as thalassemia. All three are, students, what? Autosomal. We also know hemophilia, students. We also know hemophilia and color blindness are what? They're what? They are sex linked. So if you know the table, students, if you know the table, okay. In Dharwad, nice. Hubli Dharwad, nice, students. Right. So if you know the table, this becomes easy or not, students? Tell me yes or no. If you know that table, this becomes very easy. Yes or no, students? This becomes very easy. So that is the importance of the table. Okay. Importance of the table, students. Important table. Now, the first disorder, students, how many of you can see the colors here? How many of you can see the different colors? Raise your hands. This is the color blindness test. This is a color blindness test. I want all of you to tell me how many of you can see the colors very differently. You can see red here, green here very clearly. It's a color blind test for all of you, students. How many of you can see very well? Okay. Now, if you cannot see this very clearly, very well, and if you see this by any chance, it means you are colorblind. It means you are colorblind. Colorblind people cannot distinguish between red and green. They cannot tell. Now, students, try to imagine a person is driving the car. They're going. Signal is there. Red, green. They can't tell. They cannot tell which is red color, which is green color. Gone. Okay. So, right. So, it is very important to see red and green. Right. That is called as color blindness. That is called as color blindness. Color blindness is influenced by a mutation in a gene because of which the cones and rods in the eyes gets damaged and they are not able to perform very well. Okay, it is a sex-linked recessive disorder. Here, if you go back here, students, color blindness is sex-linked recessive disorder. Sex-linked recessive disorder. Recessive disorder here. Sex-linked recessive disorder. That is color blindness. That is color blindness. Okay. Defect in the either red or green cone of the eye, unable to discriminate between red and green. Students, if this is a human eye, students, if this is a human eye, this is the background tissue of it, right? In this tissue, there is something called as rods and there is something called as cones. Now, these cones gets damaged. If these cones gets mutated, basically because of mutation, right, they cannot tell what is what. Students, what is my name in the chat? Students, what is my name in the chat? And can anyone tell me what is my name? What is my name in the chat, all of you? Right. Now, it is a defect due to the mutation in a gene present on the X chromosome. So, if you look at the karyotype, this is the karyotype right now. In this karyotype, you can see in this X chromosome, there is a defect here. Mutation on the X chromosome. Either on this, both can be affected, or in the case of male, this X can be affected in the case of male. In the case of X, it can be affected here. Okay, so this entire X gene can be affected here. Now here generally what happens, males suffer more. Why? Because we only have one X chromosome. Students, we only have one X chromosome. If that becomes defective, gone, we are colorblind. But in the case of female, students, in the case of female, there is two X chromosomes there. So in the case of female, even if one is affected, do they have a backup? They have a backup there. So in the case of female, they generally do not affected here. Okay. Students, do you want to know a fact? In the Y chromosome, there are around 75 genes. On a Y chromosome, there are 75 genes. Do you know how many genes are present on X chromosome? On X chromosome, there are almost 900 to 1400 genes on the X chromosome. That's why X chromosome is here sometimes. Okay. X chromosome is there. X chromosome has so many genes. Okay. Now, students, now in examination, what they will ask, students? In examination, what they will ask you? Okay, students, let me just open my book. I have my Tattva here, students. All of you have Tattva, right, students? All of you have Tattva books. So I'm taking a question from a Tattva book, okay? Now here is a question from Tattva book. Let's take a question here from Tattva book here. Okay. Now question says that this is how the question will come in examination, students. This is how question will come in examination. If a colorblind man, marries a girl with normal vision, okay, normal vision, what percentage of offsprings are colorblind? Okay. Now, how do you solve this question, students? How do you solve this question? Very simple. Okay, students. Students, how do you solve this, students? How do you solve this question? You will get these notes on Telegram. Okay. Telegram, I'll upload them directly. Okay. Now, students, colorblind man means what? Person is colorblind. Color C on top. Normal vision means what? Normal vision is this. Let's do a cross. XC, XY, and we have here XX. So, what do we get? Offsprings. XC, X, XY, XC, X, and XY, students. Yes or no? Students, yes or no? So, percentage of offsprings being colorblind is how much percentage? Tell me, students. See, this person is carrier. Normal. So, 0% chance that someone will be colorblind here. Yes or no? 0% chance that someone will be colorblind here. 0% actually. Students, these two are carriers. See, she has ladies can be carriers here. Women can be carrier. When both are present, they'll become colorblind. But right now they're carriers. So answer is 0% actually. Okay. Students, do you want one more question? All of you? One more question. One more question. Okay. Write down, students. Write down. Write down, students. Write down. Okay. What will happen if a carrier woman marries color blind man? Color blind man. Percentage of male affected now. Male affected. Percentage of only male affected. How many males are affected, students? How many males are affected? Okay. समझिए इसको. Understand, students? See, carrier woman is what? Carrier woman is what? XCX. She's carrier right now. Carrier. Okay. Colorblind man. XCY. So what do we get here? Okay. Now, let me teach you, students, what type of question can come in examination. Now the students, now the question says percentage of male affected. So we do not take this at all now. We forget this part. Female we forget only, because question is asking only male now. Now this entire set becomes 100% now. Out of this 100%, how many are affected, students? Tell me. Out of this 100%, how many males are affected? 50% of male are affected. 50% of male are affected, students. 50% of this males are affected here. Now, students, I'll change the question now. Let's say I'll change the question to percentage of offspring affected. Now, in that case, this entire thing becomes what? Entire thing becomes 100%. If I say offspring, all four should be considered. In that case, this is affected. This is colorblind person, carrier, normal. So in this case also, 50% of offsprings are affected. 50% of offsprings are affected, students. In this case also, see, colorblind. So entire thing is 100%. So students, divide this entire 100% like this. 25% here, 25% here, 25% here, and 25% here. Okay, 25% here, students. Okay. Now the question is asking you, students, percentage of offsprings affected. Okay. Offspring affected 25%. 25% affected. Totally 50% is affected, students. Totally 50% is affected. Ankit, Kotrish, understand? Offspring. I'll change the question, student. I'll change the question. Okay, I'll give you one more question. NEET PYQ. Here's the students. Here's the NEET PYQ for all of you. Here's the NEET PYQ. Question. See, read the question very carefully. It says that female, colorblind female I have. Okay. XCC, colorblind female. She marries a man whose mother was also colorblind. Now she is marrying a man right now. Man. Okay. This is the man here, man, whose mother was also colorblind. His mother was colorblind. So mother was colorblind here. So if this mother was colorblind, can he get this X chromosome from here to here? Yes or no? So he can inherit this X chromosome from here to here. Okay. So he gets the C. He also gets the C. So in that case, this person also becomes colorblind. Yes or no? This person also becomes colorblind right now. Okay. Let's do a cross now. Students. The question is asking you, what are the chances of her progeny is having colorblind? Okay. Let's do a cross. XC, this is the female here. XCY is the male. So what do we get? Both are colorblind, and colorblind also. Students, tell me, what percentage of the offsprings are colorblind right now, students? All of you. 50, 25, 25, 25, 25. Entire 100% is colorblind here. Entire 100% is colorblind. Okay. So usually, how can female be colorblind, students? Female can be colorblind if father is colorblind and mother is a carrier. In that case, it can become colorblind, students. Students, here's one more question. Okay, one more question. Last question. We'll go front after this because it's very important for all of you to understand the type of question you can expect here. You will not get easy questions. You will get this type of questions in examination. The question says, very simple, students. Very simple question. It says color blind. Okay. It says color blind man. Okay, color blind man. X C Y. Okay, I'll do the cross. I'll do the cross directly, students. Cross. XCY, colorblind man, marries a woman who is carrier for colorblind. She's a carrier. So only one C will be carrier, one will be normal here. Let's do a cross. XC, XC, XCY, XCX, and XY. The question is asked. Now let's see the question now. Okay. It is X-linked, not Y-linked. It's X-linked disease. No, it's X. This entire disease is because of mutation on the X chromosome. Mutation on X chromosome. Yes or no? Let's see. The question is asking what is the probability of their children having color blind? It says children. So color blind, carrier, carrier normal. So in this case right now, 50% is color blind. Yes or no, students? 50% is colorblind right now, not 75. How is 75, students? This person is carrier. For a female to be colorblind, it should be both. Both X chromosomes has to be affected for female. But for male, only one is enough. So 50% are colorblind here, students. 50% are colorblind. 50% are colorblind right now. Okay. Now, students, next is called as hemophilia, students. Next is called as hemophilia disease. Next is called as hemophilia, students. In hemophilia, very simple, students, very simple hemophilia. One small cut, blood keeps on flowing. One small cut, blood keeps on flowing. That is, in the case of hemophilia, a person will have, with a small cut, will keep on bleeding because the clotting protein is missing here. Okay. That is, sex-linked recessive disorder. First of all, hemophilia, all of you know, sex-linked it is. A single protein that is part of a cascade of protein involved in the blood clotting is affected. So that blood clotting is not happening here. In this person, the blood clotting is not happening because the clotting factor is missing here, students. Okay, blood clotting is missing. Okay, now students, now here your family pedigree of Queen Victoria shows the number of hemophilia descendants. She was a carrier for disease. So Queen Victoria has the hemophilia. Now again, students, this is also X-linked here. Hemophilia is also X-linked. Generally, here males are more affected compared to females again. Now we'll do a question here to get a better understanding. The question is here, students. See the question. Read the question. Very beautiful question it is. It says, Mary's father has hemophilia. Let's take, we have Mary here. Mary is a girl. Okay. Mary's father has hemophilia. Father has the hemophilia. So this X chromosome, let's say mother is normal. For now, let's say mother is normal here. Let's say mother is normal because information is not given. Assume normal. So this X chromosome came here to Mary. This X chromosome came here to this Mary. Okay. Now, husband is hemophilic. Husband is going to be hemophilic in nature. Husband is hemophilic. Husband is hemophilic in nature. What would be the chance other daughter having this disease? They're asking you, what is the chance that daughter being diseased now? Okay, let's do the cross. XH, because she calls X chromosome from here, from the father here. Okay, and here we have XH and Y. X, H, XX, XH, XH, XY, XHY. Now students, here we can see a carrier female. Here we can see normal son, hemophilic son, and hemophilic daughter. Now, if you look at these options here, that they have asked daughter. So we have to technically forget about this entire part. Forget this entire part, male part. Now, out of this 100%, can I tell 50% is affected, 50% is normal here? Yes or no, students? Yes or no? In this case, 50% are affected here. 50% are what? 50% are hemophilic. 50% is carrier. So, what are the so this has to be 50%. This has 50% has to be answer. Not 25. 50% has to be answer. Okay. What would be the chance of daughter having disease? Daughter is 50%. 50% is the answer technically. Okay. 50% the answer. Not 25. 50% has to be answer. Okay. So you'll get such questions, students. You'll get such questions. I'll give you one more question from my Tatva here. Okay. I have one question here, students. Let's say we have a carrier female. Carrier woman marries normal man. Okay. Tell me percentage of offspring hemophilic in nature, students. Okay. See, carrier woman is what? Carrier woman has to be XH, X. Normal man has to be XY condition. What did we get here? XHX, XX, XHY and XY. Tell me, students, what percentage are hemophilic here, students? Tell me, students, what percentage is hemophilic here? All of you. See, this is your carrier. Normal. This person is going to be hemophilic in nature. So 25% of the offspring, 25% students, not 50, Ravi. 25% of the offspring are hemophilic here. 25% are hemophilic, not 50. 25%. 25% are hemophilic. This is carrier. Normal. 50% are normal. 25% are carrier. 25% are hemophilic. Understanding? 25% are hemophilic, students. 25% are hemophilic in nature. Now, students. Now, next is called sickle cell anemia. We have two more diseases. We are over after that, students. Sickle cell anemia, what happens is that the shape of the RBC changes. The entire shape of the RBC changes because there is a point mutation. There is a point mutation, right? A point mutation. Okay. So, it is an inherent red blood disorder. Autosomal linked recessive disorder, students. It is an autosomal linked disease. Okay, autosomal linked disease here, right? Now, students, can you see the disease? What happens in the disease condition? In disease, there are three categories here. We can have normal person will have two normal hemoglobins. Carrier will have, obviously, one is your sickle cell anemic. For sickle cell anemia to become, for sickle cell anemia to happen, both should be affected. Both alleles should be affected here. Okay, now students, see this. How does it happen? Now, in normal hemoglobin, what is it, students? Normal hemoglobin, right? We have GAG here. Now here this adenine is replaced by GTG, by thymine. So adenine is replaced by thymine here, you can see, students. Now because of this 1 point mutation, can we see the mRNA changing here? GAG become GUG. Now if mRNA is changing, the hemoglobin peptide chain is changing. That is, in this beta globin chain, in the 6th position, in normal condition, we have glutamic acid. Glutamic acid is replaced by valine here. Because of this 1 point mutation, the entire chain is affected here. So here we have glutamic acid is replaced by valine here. Okay. Change in the amino acid sequence is caused due to the point mutation. The glutamic acid in the 6th chain of the beta globin chain is replaced by valine. So we get valine. And because of this of point mutation, because of this 1 point mutation, the shape of the RBC becomes sickle shape. It becomes sickle shape here, sickle shape. And it can make sure there is, because of this hemoglobin now shape has been changed, so it cannot carry oxygen anymore. Properly. Okay. Now in examination, you also can get questions. You also can get numericals. See this numerical here. It says A cross between male and female, both are students what? Both are heterozygous for sickle cell anemia. Both are heterozygous. So HbA, HbS. HbS. This is male. Okay. Let's take your this is male. This is female now. HbA, HbS. Both are heterozygous condition. The question is asking you what percentage of progeny will be deceased. Let's do the cross, students. Let's do cross. HbA, one child will be normal, one child will be carrier, one more child will be again a carrier, but one child, students, HbS, HbS, this child is going to be sickle cell anemia, students. This child is going to be sickle cell anemia. So answer is what? 25%. Can you see? Entire thing is 100%, 25, 25, 25, 25. 50% are carrier, 25% is normal, 25% is sickle cell anemia. Sickle cell anemia, students. Sickle cell anemia. Students, are you able to follow this amazing concept? How the question will be asked in examination. That is the beauty of what I teach. I make sure you learn what is present, what is important for exam point of view. Exam point of view, students. Exam point of view. See, students. In disorder in which a single change in base pair, that is your sickle cell anemia. Okay, students. Now, what exactly happens in the case of phenylketonuria, students? In the case of phenylketonuria, understand. In the case of phenylketonuria, what happens is that in normal condition, phenylalanine gets converted to tyrosine. In normal condition, phenylalanine gets converted to tyrosine because the enzyme is present. Which enzyme? Phenylalanine hydroxylase enzyme. Phenylalanine hydroxylase enzyme is present here. Is present. Okay. Now, in disease condition, what happens? In disease condition, phenylalanine is not converted to tyrosine because the enzyme is missing. The enzyme is missing right now. If the enzyme is missing, what happens? Phenylalanine is accumulated and converted to phenylpyruvic acid. And this phenylpyruvic acid starts to accumulate in the brain, starts to accumulate in the bones, start to release as in the urine also. In the urine also, students, urine also. So because phenylalanine is not converted to tyrosine, phenylalanine gets converted to acid, that gets accumulated. You see, accumulation of these in the brain can cause mental retardedness, excessive through urine because of poor absorption by the kidney. Because of poor absorption of kidney, students. Class 10 minutes will be over. Okay, 10 minutes. That is your phenylketonuria, students. Next, what is thalassemia, students? Last, this is of Mendel. Mendel's last disease. Mendel's last disease called as thalassemia. Thalassemia, again, students, mutation. Mutation only. Here you have to remember 2 points. I'll tell you what you have to remember, students. Okay. Again, it's a blood disorder. Blood disorder, students. Blood disorder. This is a blood disorder similar to your sickle cell anemia. Okay. Blood disorder resulting from inadequate HB synthesis. That is, hemoglobin is not produced here. Hemoglobin has a defect here. See, students, if you know the structure of hemoglobin, this is the structure of hemoglobin. In a hemoglobin structure, there are 2 alpha chains and there are 2 beta chains. 2 alpha chains and 2 beta chains in hemoglobin. Okay. Now, what exactly happens in the case of thalassemia? In the case of thalassemia, there is a mutation. There is a mutation, students. In alpha thalassemia, what happens? HbA2 gene and HbA1 gene gets affected. These 2 genes are present on the chromosome 16. Now, because of this mutation in these 2 genes, alpha chain is not produced. Here, in alpha thalassemia, alpha chain is not produced. So, alpha thalassemia happens. Now, in the case of beta thalassemia, students, in the case of beta thalassemia, beta chain is not produced. Decrease in beta chain. Why? Again, a mutation on chromosome number 11. Because of a mutation on chromosome number 11 in HBB gene, beta chain is not produced. So please understand, students, here chains are missing. So it is a quantity problem. It is a quantity problem. Right? Alpha chain is missing, beta chain is missing, alpha thalassemia, beta thalassemia. So it is a quantity problem. But in the case of sickle cell anemia, there it was a quality problem. So in the case of sickle cell anemia, it was a quality. See, here valine came because of glutamic acid, valine came. Now valine came there, so quality of the chain was missed here. Quality of chain is gone. But here chain only is missing. Chain only is missing. This is the quantity. Sickle cell anemia is a quality problem. Quality problem. Okay, students. So you can get such questions here, students. Sickle cell anemia and hemophilia are autosomal, students. Are both autosomal? Students, tell me. Students, is hemophilia autosomal disorder? No. Hemophilia is sex-linked. Hemophilia is sex-linked. Okay. Sickle cell anemia, both are disorders of blood. Both are disorders of blood, students. Okay. So this is incorrect. This is correct. Hemophilia is sex-linked. Hemophilia is sex-linked. Again, your sickle cell anemia, students. Sickle cell anemia is also recessive. Sickle cell anemia is also a recessive disorder, right? Recessive disorder. Not autosomal dominant. It is not autosomal. Autosomal dominant, we only have what? We only have myotonic dystrophy. Myotonic dystrophy, students. Okay. Now, students, last three disorders of chromosomal disorders. In chromosomal, we have Down syndrome, Turner syndrome, Klinefelter syndrome. In Down syndrome, what happens, students? In Down syndrome, can you see this chirotype? In this chirotype, 21st position, can you see one extra chromosome here? All of you. In this chirotype, can we see one extra chromosome 21st position? Yes, we see. So in a Down syndrome, what happens? In Down syndrome, what happens? It is also called as trisomy of 21. Why? Because in the 21st position, there is an extra chromosome. Aneuploidy. Aneuploidy is happening here. Aneuploidy is happening. It occurs both in males and females. First discovered by Langdon Don. Okay, one extra chromosome is present in 21st position here. Okay. Here, symptoms is very important. Symptoms is very, very important, students. Symptoms. Okay. Symptoms, students. What are symptoms here? Short stature. Persons are very short. Students, have you seen people like this in TV or somewhere? Have you seen such people? This is because of Down syndrome. This is because of Down syndrome right now. Short stature, right? Small round head here. Furrowed tongue is outside, right? Partially open mouth. Palm is broad. Palm is very broad with lot of lines on the palm here. That is palm characteristics. Physical, psychomotor and mental development is not seen here. Not seen. So all of these are very important. All these points are very important. Apart from this, diagram also they can ask you. So please learn the diagram also. Palm creases, many loops on fingertips, flat, big and wrinkled tongue, congenital heart disease is there, congenital, broad, flat face. This you have to remember, students. Okay, this is what happens in Down syndrome. Now, in the case of Klinefelter students, Klinefelter syndrome is seen only in the case of males. Only in the case of males. In Klinefelter syndrome, there is a, see, normal male is what? XY. In the case of Klinefelter syndrome, XXY condition. XXY condition. There's one extra X chromosome here. Extra X chromosome is seen here. Okay, extra X chromosome. Now, because of the extra X chromosome, what happens? Symptoms. See, such individual has overall masculine development. However, feminine development also happens. Feminine development, students. Feminine development here. Here you can see development of breast. Development of breast is called as what? Gynecomastia. It is called as what, students? Gynecomastia. Gynecomastia. Okay. Gynecomastia, students. Gynecomastia. It is also expressed, such individuals are sterile in nature. Gynecomastia is seen here, students. Now, Turner syndrome, students. Turner syndrome. Turner syndrome. Okay. Counselling, I'll teach, students. Counselling also will make videos. Okay. In Turner syndrome, what, students, happens? Turner syndrome is only seen in the case of female. Only seen in the case of female. Now, in normal female, it is XX. But in Turner syndrome, one X is missing. One X is missing. And because there's one missing X here, can you see? Missing X here, X chromosome here. Such females, sterile, ovaries are rudimentary, besides other features including lack of secondary sexual characteristics missing completely. That is what basically happening in the case of Turner syndrome. With that, chapter is over. Students, with that, this is the Turner syndrome again, students. Different features are given here, given to you, students, given to you. With that, chapter is over. Here are some questions. Manifestation of phenylketonuria are given below. Choose the incorrect statement. Mental retardedness, skin rashes. Yes. Furrowed tongue. Furrowed tongue is seen in the case of Down syndrome. In the case of Down syndrome. NEET PYQs. Broad palm creases. Palm creases, that is seen in the case of Down syndrome. In the case of Down syndrome, students. Down syndrome. Okay. Now, students, let me give you a homework question. Homework question is very simple. Who gave the chromosomal theory of inheritance? Okay. No. Who gave the experimental proof? Students, who gave the experimental proof? Okay, experimental proof on your fruit flies. Fruit fly. That I want in the comment section. That I want in the comment section. The answer in the comment section. Students, chapter is over. Right. Students, this detail I also go in DNA batch also. So if you're not a part of DNA batch, please join, because there I am teaching right now. In DNA batch right now, I am teaching chapter called as your morphology of flowering plants. Morphology, I'm going in so detailed there. Next I'll be teaching anatomy there. Anatomy also. Right. So students, this I want in the comment section. All of you comment section the answer. Fruit fly is called as Drosophila melanogaster. Fruit fly is called as what? Drosophila melanogaster. Which scientists worked on that? Students, I will look forward to all your comments in the comment section. So Jane, Jenny, Adina, Science World, Ravi, all of you, from starting till the end, I look forward to your comments in the comment section. So thank you so much, students. Bye bye. Take care, all of you, and have a great day ahead, all of you. Bye bye. Thank you so much, students. Last slide of mine. Thank you, students. Notes will be given in Telegram. Download notes from Telegram channel, Vedantu NEET English Telegram channel, as well as Basavaraj Sir Biology. You get the notes there. Okay, students. Bye bye, students. Bye bye, all of you.